Now that we have built models and we've narrated those different video clips, then you probably don't need me to label the different parts of this diagram, but let's review and see what you remember. Remember that tRNA molecules that you see here have 3 bases on them, and you should be able to tell me the 3 bases are called an anticodon. So there's 1 anticodon per 1 tRNA. You need to be able to tell me that on the mRNA, every 3 bases is considered a codon. So let's just go ahead and frame off the first 3 right here and label them the codon. You should be able to tell me that each one of these dots represents the monomers of proteins, which are called amino acids. So that's an amino acid. You should be able to tell me that the line between each of those amino acids is representing that bond, that is that peptide bond. We have the ribosome here, which is assisting in matching those tRNAs with their anticodons to the codons on the mRNA, bringing the amino acids for the proteins. Now there's several sections in our notes that we're skipping through, and that's because we'll just come back and we'll use them as warm-ups as we finish up the unit and get close to test time. We can take a second to look at this. This is just kind of a different way to ask you questions about transcription and translation. They're giving you one side of DNA, AAT. So if I ask you to identify the mRNA codon, you should be able to tell me it would be UUA. And this process of going from DNA to mRNA was transcription. On the test, I feel like it's really important to emphasize that when we say identify a process, you should be talking about transcription or translation or protein synthesis is a process. If I say identify a molecule, then you should be listing things like DNA and mRNA and tRNA. If I say identify an organelle, then you should be listing things like ribosomes. So it's important that you understand kind of the categories that these different vocab words fall under. So when we take our mRNA, we're going to hook those tRNA anticodons up. So the codon that would be complementary to the mRNA codon would be AAU. And the tricky part is trying to figure out our amino acids, because we commonly go right to the tRNAs because they're right next to this amino acid. But we need to remember that to figure out this amino acid, we have to go over here and let's use the mRNA, because if you use the tRNA, you're going to mess up your amino acids. So if we go and look up UUU, then that's going to give us the name of a specific amino acid. Now I want to get into something that you already know. And to review this concept, I want to show you a video clip that you probably remember seeing when we were in the cell unit. And this is kind of putting it all together here. We're going to see some transcription and translation happen, and we're going to make a protein at the ribosome, but then you have to remember what then could happen to this protein. Because sometimes ribosomes are on the rough ER, and sometimes ribosomes are just unattached and they're in the cytoplasm. And they're both going to make proteins, but they're going to make proteins for different parts of the cell. So we see coming out of the nucleus, that red little line there, that's the mRNA that was made from transcription. And the mRNA is on its way to a ribosome. And that little black dot right there that you see, that's a ribosome that is attached to this other structure. You probably remember that is the rough ER. And you can see that the ribosome has grabbed onto that red piece of string there. That's the mRNA, and it's translating it, and it's making a protein. And we're going to see this protein that it made is getting stuffed right into the lumen, or the inside of the rough ER. That protein folded up, and we see the protein is now going to be sent to the next organelle, which is the Golgi. The Golgi is going to further modify and tag these proteins for their final destinations. And you need to remember that proteins that were made by those attached ribosomes that we just saw, these proteins are ultimately going to end up in 3 different destinations. You're going to see that this first vesicle that we see moving away there, it's got 2 proteins in it. It's got green proteins and it's got blue proteins. And this vesicle is heading to the membrane. We're going to see the green proteins are getting embedded. So we need to know that some proteins can be embedded in the membrane, and the blue ones were secreted to the outside of the cell. So that would be like insulin, because your cells make insulin and then they dump them out. Now you're seeing a lysosome. And lysosome, remember, digest molecules, and it needs enzymes or proteins to do that. And the Golgi can tag some of these proteins to become lysosome proteins or enzymes. So you need to remember, let's add to our notes here, that if you have attached ribosomes, and I know this is a little bit of a difficult image to look at, but this would be an example of attached ribosomes. You see them right there on the rough VR. And this one has mRNA, and it's making the protein. So let's make sure you understand that here's the protein that was being made. The protein got put right into the rough VR. The rough VR has already modified it. It's sending it to the Golgi. And that we can see that these proteins can end up in 3 different destinations. Lysosome, they can be secreted outside the cell. That's what you're seeing right here by number 2. And then some of them can be embedded in the membrane. So these little spiky proteins there are becoming a part of the cell membrane. So they could be like transport proteins and receptor proteins that you learned about in a previous unit. So let's indicate attached ribosomes make proteins for, say, secretion. That means they're being released out of the cell. Some proteins are going to end up in lysosomes. And some proteins are going to be embedded. Let's put embedded in the membrane. When we look at this box up here, this is showing you our unattached ribosomes. They're not hooked onto the rough ER, and they're making a protein. You can see one being made right here, and that protein can just stay out here in the cytoplasm, because this is just saying we have the cytoplasm that the mitochondria is in. So unattached ribosomes can make proteins for the cytoplasm, and the cell is going to use them to assist in different reactions. And then we can see some proteins, like this one right here, number 2, is making it into this organelle. So some of these unattached ribosomes are making proteins for organelles of the cell to use.